this is Paul Nelson, and we're going to be, and his talk is on taxonomically restricted genes. We're starting at 11:40, so your QA time should start at 12:10.
This is a project that is being funded by the Discovery Institute, and we have an international group of collaborators working on it. I'll come to that at the end. But I think this question of how pervasive are taxonomically restricted essential genes is one that intelligent design is uniquely positioned to answer. And I'll come to my reasons for saying that in just a moment. But let me start with an analogy from art. So here is a painting from the early 20th century in a conventional academic style, showing a doctor treating an ailing woman. Here is a sculpture from the mid-20th century showing a baboon with the face of an automobile cradling a child. Here's a Cubist landscape, and here is a massive sculpture in downtown Chicago that I pass by frequently. These are all the work of one person, Pablo Picasso. Now, Pablo Picasso, as an artist, had an incredible range of output. He worked wherever he could in a whole variety of visual media. So take that image of a creative agent and counterpose that to a templating process that's deterministic, where you're working from, this is a coin stamping machine, where you're working from a common pattern without elective agency or causal freedom. Those expectations, we can think of this as sort of the metaphysical background, those expectations will give you very different possibilities for what can be represented and what can be actualized. So I like parallels between natural science and art. I think they're illuminating. And this one we'll come back to. I put it up here at the beginning so it won't seem excessively weird when I come to it at the end. So let's now segue to biology and this templating expectation. Here is the classic universal tree picture of life with Luca, the last universal common ancestor, at the singularity at the base of the tree. Now, we'll use this picture to generate expectations about essential genes and proteins throughout the whole tree. So keep this geometry in mind as we go forward, because we're going to use it really to project expectations into gene and protein space, asking if this geometry is true, what should we expect to find? Now, evolution, contrary to I think what many people believe, is not an unconstrained process. So if we start with Luca and some unknown pathway leading to Luca, we will have in Luca approximately 300 proteins, dozens of large RNAs, a whole bunch of associated molecular apparatus that is necessary for function. And take any bacterial cell that you care to choose, like E. coli or mycoplasma, you can subject that system to perturbation and really come up with a basic set of hardware that you can't do without. So let's assume that something like that basic set of hardware is present in Luca. Now, evolution says keep whatever works by necessity. In other words, essential genes and proteins are those pieces of hardware that the cell cannot do without. And evolution, as I argue in this slide, is a constrained process. So whatever works in Luca and in the sense of is essential for function will be kept by its offspring. And then this allows us to generate predictions about what we should see as we move up into the three-domain geometry of life. So we should expect to find that essential hardware in the branches leading from Luca up into the tree, and the constraints are very tight. Viability constraints on any evolutionary pathway require that essential functional elements be retained throughout all branching lineages. And what one doesn't expect then from this picture is that those essential elements are going to be replaced. All right, so Now, the way to think about this is there's no dying in evolution. There's no crying in baseball, and there's no dying in evolution. Viability and reproductive capacity must be maintained along with any cellular or organismal features necessary for those functions. So, Luca, present here, gives us expectations about what we should see as we move through the rest of the tree. So here's a photo of my desktop. This is actually, this phone is just to my right here. And that spiraling cord is supercoiling on itself because of its geometry. So probably once a week I have to disconnect the cord from the handset, let out the supercoiling, relax the cord. Otherwise I end up with one big knot and I really can't lift the handset up to my head when someone calls me. So here's our problem. We have got a spiraling geometry that is circular, and if we don't relax the supercoiling, we're going to have functional problems. DNA, the geometry of DNA creates the same functional difficulty for any cell, and that is managed by proteins called topoisomerases, as many of you know. They fall into two major classes. Topo 1s make a cut in one of the sugar phosphate backbones of the DNA double helix. Topo 2s cut them both. And they are demonstrably essential for cell function when you have a DNA genome. So Luca, by hypothesis, possessed a DNA genome. Therefore, it should have a functional Topo 1 and a functional Topo 2. And again, on the basis of our starting premise that evolution is not unconstrained, we should expect to see ubiquitously throughout the remainder now of the tree, Topo 1 and Topo 2 present as we move through those different groups. So what do we find? Well, this is an example I've often presented to audiences because it's so dramatic. In an archaean, Methanopyrus that lives at temperatures that would be very uncomfortable for us, very high temperatures, that archaean possesses a topoisomerase that is found only in that species. As far as I know, I checked this recently, the taxonomic distribution of this topoisomerase is present just in that group. Now, when this was discovered, it caused some head scratching, understandably. And the French investigators looking at this then moved out through the whole of the tree, looking at the distribution of other topoisomerases, asking the question, if common ancestry is true, what should we expect to see? Now, from their paper here in 2009, they begin with what I think is a rather quixotic take on the question. They say, we would expect from Luca one Topo 1 and one Topo 2 distributed ubiquitously throughout the tree. But look how they phrase it. An intelligent designer would probably have invented only one ubiquitous Topo 1 and one ubiquitous Topo 2 to facilitate the task of future biochemists. Reality has turned out to be quite different and more interesting. The distribution of these essential enzymes is not congruent with the universal tree based on 16S ribosomal RNA. So here's a figure from their paper, and you can see that the diversity of topoisomerases throughout the tree. Now, the tree here is represented. Here is the root at Luca. We have the eukaryotes here, the bacteria here. The three major groups of the archaea are here. There are so many different topoisomerases that this French group invents ancient viral reservoirs to give rise to them. We need to realize there's actually no there's no direct evidence for these ancient viral reservoirs. They are postulated deus ex machina to provide a source for these functionally the same. I mean, they're doing the same job. These are DNA disentanglers, but structurally very, very different enzymes. Another example of this sort, as many of you know, transfer RNAs are highly modified by enzymes. In particular, this base 3' to the anticodon is methylated in many tRNAs. That function is performed by a methyltransferase, and this this enzymatic modification, methylating this G here, is present throughout all of the three domains. It happens in the archaea, the bacteria, and the eukaryotes. So your expectation would be, again, based on this geometry of universal common ancestry, that LUCA would have these modified tRNAs, and it should have that enzyme. It should be present here. So our straightforward expectation is, based on this geometry, that we're going to find that same enzyme present throughout the tree. Here it is. Well, here are two versions of it. This is the bacterial enzyme here. This is the archaeal enzyme here on the left. They are not homologous. They're fundamentally different enzymes. And in this paper in JMB a few years ago, the authors point out that this methyltransferase is not, contrary to expectation, homologous throughout the tree. As they put it, the invention of two distinct enzymes. I don't think they looked at eukaryotes. You might find something different there. I'll have to pursue that in terms of more recent publication. They say the invention of two distinct enzymes to catalyze that reaction was unexpected, given that the modification is seen throughout the tree. This demonstrates what they call the principle of non-orthologous gene displacement. This is a name, this long phrase is a name for a mystery that I think is going to be increasingly amplified as whole genome sequencing pours more and more data into our labs. One last example of this sort, termination of protein synthesis in all three domains of life is catalyzed by release factor, which comes in as the stop codon enters the ribosome. At the A site, release factor comes in, hydrolyzes the bond between the tRNA and the P site and the last amino acid of the polypeptide chain, releasing the protein to go off and fold. Again, release factor has to be there. It's an essential enzyme for protein synthesis. So what would your expectation be? Again, based on this geometry, LUCA has a ribosome. It should have release factor performing that job. So here is the crystal structure of release factor in bacteria. And what would you expect to find in eukaryotes? You'd expect to find, on the basis of that tree and LUCA, a homologous enzyme. In fact, it's totally different fold. There's the only similarity between these two proteins is in the active site, 3 amino acids. Everything else about the proteins is different. So when this was discovered, again, the authors did some head scratching. They said the evolution of known well-established class 1 release factors holds unsolved puzzles. There is no strong evidence of an evolutionary relationship between bacterial class 1 release factors and their counterparts from archaea and eukaryotes. Ergo, it is unknown how termination was mediated in the last common ancestor. So we see a pattern here where we come to the tree, the three major domains, with expectations based on that singularity of LUCA, only to have them overturned as we examine particular cases. So I think, in general, this problem falls within a remarkable discovery of the last 2 decades, and that is orphan genes and proteins, mediated by a technology. This, when I was a graduate student, none of this was known because we simply didn't have a sample size large enough to catch these anomalous patterns, but automated DNA sequencing now has provided that, and the incredible growth of data. So here we see, this comes to 2008. I was a graduate student back here. Really, prior to that, prior to the DNA sequencing revolution, we knew basically nothing about the genetic diversity of life on this planet. We had a tiny and unrepresentative sample of what was out there. Bring that figure more up to date. Here we come now to 2014. When you have a curve like this, and this enormous accumulation of novel information, it's bound to upset your theories, and I think that's what's happening right now. So as some of you know, Richard Buggs and I published a chapter last year with Cambridge dealing with this, and in their editorial introduction, Peter Olson and his co-editors of the volume said, Nelson and Buggs introduced one of the most fundamental unanswered questions raised by this deluge of genomic data, the presence of orphan or taxonomically restricted genes throughout the tree. Every genome thus far has revealed an unexpected percentage of unique genes, and increased sampling of gene space has resulted in this space expanding rather than contracting as would be expected if genes only appeared to be orphans due to lack of sampling. And then they point out when these genes are examined, they turn out to be functional. They're not baggage. So as some of you know, in December, Richard and he was the senior author of a publication in Nature with a very large group of collaborators looking at the genome of the ash. There are some species of ash that are resistant to diseases that take down other species, and so Richard and his collaborators were looking for the genes and proteins present in the resistant species to find out how they were surviving when other species were being taken down by diseases like ash dieback. When that paper appeared, Richard published an accompanying piece in Nature, Ecology, and Evolution with the title The Evolutionary Mystery of Orphan Genes, where he points out, as you can see here in the subtitle, every newly sequenced genome contains genes with no traceable evolutionary descent. I believe in the ash genome it was about 25% of its genome was orphan sequences. So I'm well known as a sort of evangelist for this, or an enthusiast, a cheerleader for orphans, and that's, I think, for good reason. I think these findings are really going to challenge our expectations based on existing theory. So what are orphan genes? Well, very quickly, you have newly sequenced data that's been fed into some gene finding program, and there are candidate open reading frames in that sequence. You subject those, so that would be at the level of species or genus. You run those into a genomic database, and you use a homology threshold, typically 10 to the minus 3 to 10 to the minus 5 expect value in BLAST. That gives you your protein coding sequences found only in one species or genus through that pipeline of that analytical pipeline. Now, later in the talk, I'll mention some sequences that are not protein coding, but nonetheless are orphans, and I think are equally interesting. But let's focus just for the moment on protein coding sequences. Known sequences, those stored in GenBank, have been annotated and deposited there with respect to function. So one of the first indicators that you've got an orphan is you have an open reading frame of unknown function. Because if it had a function, it would already be deposited in GenBank with that associated function, and you would know what it was doing, what that gene and its protein product were doing. So here we have a match, some high degree of similarity as you go down the reading frame. This is a ribosomal protein that tells us what our newly sequenced gene is doing. With orphans, however, we find no matches. And you can vary the expect parameter in BLAST, and the signal is remarkably consistent. So here's our orphan. It's got the characteristics of a protein coding sequence, but it finds no matches. So a parallel, excuse me, there are helpful parallels that we can draw with natural language like English. Pretty much any English novel that you look at, piece of fiction writing, will have these words. These character strings will be present universally. Then we have what we could call the mid-range words, which would not be present universally. You can probably guess what this story is about. And then you have the uniques. Okay, these are the words that only occur once. You never find them elsewhere. Now, I had to make these up, okay, because it's extremely rare in English for a word to be present only in one text. Actually, this word, Meki here, is a slang word in my family that my daughters call me when they're annoyed with me. So this, in the Nelson family, is an actual word. But the rest of these I just made up because I had to have some examples of these unique words. The parallel to genetics is remarkably strong. So we have a universal or conserved core set of genes. Typically, these will be associated with ribosomal structures. These are almost invariably, these will be ribosomal proteins. Our next set, sorry, my advanced key isn't quite working. Our next set here will be those that are less conserved. Some authors have called these character genes, genes and proteins present in some environmental niche, for instance. But we find them in multiple species. The orphans, by contrast, are going to be unique. And, sorry, now it's advancing. And because of the way we count them, this bin will get much, much larger, much more rapidly than the other two. In fact, it ends up swamping, in terms of total numbers, those genes that are and proteins that are conserved. So this group in Germany has looked at this and actually come up with a very, very interesting method for classifying the distribution of orphan genes I don't have time to go into today. But as they point out in this publication, orphan genes vastly exceed the number of known gene families because of the way that they're counted. So this Israeli team, early in the history of the discovery of orphans, so we're looking now at about 14 years ago, they point out that given our background expectations, we do not expect to see orphans, given that universal tree geometry and assumptions about how new genes and proteins come about. Russell Doolittle, 2 years later, again doing a survey on the basis of, at the time, a fairly limited number of genomes compared to what we have now 11 or 12 years later, he said, we thought that we could do whole genome analysis based on really just matching algorithms, given our expectations, namely that extant genes are descended from a smaller ancestral population that has expanded by gene duplication. So the picture that I think many biologists have in their mind is if you get a change in a protein structure, here we've added an alpha helix by some internal duplication within this reading frame, that you will retain a signal of history. These folds, with the exception of this alpha helix here, these folds are the same. And these gene sequences are going to have high homology one to another, even though we've added a structure here. With that in mind, if you begin with LUCA and the singularity of LUCA with its essential hardware, you should expect to see ramifying out here through these three domains the same essential hardware being conserved. And from that perspective, orphans, especially when they're essential, turn out to be anomalies. So in the background, we have common descent as a given. That's not at issue. We think we know how new genes and proteins fold. How they arise, and they're going to leave a trace of similarity or homology that will allow us to, build their ancestral pattern. Given 1 and 2, you do not expect to find a lot of unknown genes and proteins. Now, I don't have time today to go into the functional role of orphans, but please, as a take-home message, realize that when the functional role of orphans is assessed, they turn out to be essential to the biology of their respective species. So many salamanders can regenerate their limbs. Orphans are involved in that process. In this group, freshwater polyp hydra, the very morphology of this species is mediated in development by orphan, or what this group calls novel genes, species-specific morphological traits. So one of the things you can do when you have a complete genome, this is a circular bacterial chromosome, is subject it to perturbation analysis, here by introducing a transposon which integrates randomly in the reading frames in that chromosome. If you hit essential hardware, you'll get a signal. You won't get any growth. Those cells will die. Same thing happens here. At least one of these transposon integrations has hit essential hardware. In some cases, however, the transposons will integrate and you'll be able to grow the cells. As you go and look then at their DNA and you see transposons in living cells, you know that to a first approximation at least, the transposon has not landed on essential genes because the cells are growing, right? So they've got what they need, at least in under laboratory conditions, to make a living. So this experiment has been done many times using different bacterial groups in particular, and the results are always interesting. I'll look just at one example. Lucy Shapiro's group at Stanford did this experiment with Colobacter, a bacterium you can culture out of fresh water. And they determined it had a little over 1,000 essential genomic features, including 90 segments of unknown function. Nearly 1/10 of its essential hardware is orphans. And Shapiro, when he was interviewed about this, said, we found all of this essential hardware, essential in the sense that transposon mutagenesis showed it to be demonstrably necessary for cell viability, but we don't know what it's doing. We have no idea what's going on there. And she says, well, this may lead us to new and completely unknown bacterial functions. Well, that's sort of the cell function or cell physiology lesson of the experiment. The evolutionary lesson, I think, is even more interesting. So Colobacter and E. coli are evolutionary relatives in the proteobacteria. In our three-domain picture, they share a common ancestor. Yet, as you can see from this Venn diagram from the Shapiro paper, there is a large fraction of the Colobacter genome that is essential, demonstrably essential for function, yet taxonomically restricted to that genus. If we go over here to E. coli, we find a corresponding, even larger fraction of essential elements present in E. coli, necessary for cell viability, but taxonomically restricted. If you did this experiment with another member of the proteobacteria, you would get a similar Venn diagram, some region of overlap, some region of essential, species-specific or genus-specific hardware. A result, I think, that is very hard prima facie to reconcile with that single tree picture. So I'm almost done. I'm going to finish on time. I'm so happy. So let's step back and look at this from a theoretical standpoint. LUCA, if it existed, gives us a complex vector that will define a radius for us. And the vector is defined by the functional demands of viability, known mutational processes and time. So we can put that in here. That will give us a radius. And this now is a sphere outside of which we should not expect to find genes and proteins if LUCA existed. But that sphere is going to be only a tiny neighborhood within what is a very much larger space accessible to a designer. Because a designer is not bound by any kind of material template. To go back to my original example of Picasso versus a coin stamping machine, a designer can move to any point in a very much larger space and actualize proteins. This then gives us a way of, on the basis of whole genome sequencing, distinguishing the predictions based on Darwinian universal common descent, this radius or this sphere, as opposed to what could happen under a design framework. I'll skip that. Change Tan is one of the collaborators in this project. It's called Orphan Base, which Discovery has funded. And this paper, I just think, is so unbelievably interesting because what Change has argued is it should be possible to use this flowchart and ask the question, if we have essential genes that are taxonomically restricted, it is very likely that that group does not share a common ancestor with its neighbors that do not share those taxonomically restricted essential genes. I wish I had more time to go through the whole logic of this, but I'm convinced that Change is onto something. And in the next year and 18 months, she and I and another group of collaborators will be working on this and really coming up with an automated pipeline to try to get at these sequences and find out what they're doing and how widely distributed they are. Folks, I want to tell you I have barely scratched the surface. So one of the examples I wanted to include but realized I couldn't do it in a half hour is there's a group in Australia that's looking at the role of taxonomically restricted essential genes in the mollusks. It turns out that mollusks within the phylum Mollusca, phylum-defining features like the mantle are built with taxonomically restricted genes and proteins. So as you move from one molluscan group to another, they are not building the anatomical features that define that phylum using the same genes and proteins. And this pattern appears to be quite general. In other words, it's not just anomalous in the mollusca. As you move out into other animal groups, you find the same pattern. So again, I get goosebumps because this is not something that one would expect on that monophyletic picture that I began with.
Jim Lemaster said he especially liked the decision flow diagram in that last chart, which I also thought that was great.
Yeah, I love the way Change thinks. And will pat myself on the back, which I don't ordinarily do, but Change was a student of mine at a Discovery Institute summer seminar a few years ago, and I was able to get her excited about orphans, and she's taken this and run with it. So I'm really looking forward to collaborating with her on automating this and making the signal as clear and unmistakable as possible.
Can I ask a question, Paul?
Sure.
Okay. I've been looking into this question of where we get new genes, and it's relevant to the design inference because what happens is people will say things like, well, we get new genes because there's non-coding sequence that gets converted to coding sequence, etc. What percentage of orphan genes would you say that's true for?
I would say it's true for maybe 20%. The problem is it's very hard to determine the arrow of polarity there. In our chapter, Bugs and I look at this, and given the information we have, it may be the case that the apparently non-coding sequence in a related group is actually has lost its promoters or has in some way been disabled. So it's hard to know what came first. Was the non-coding sequence the original sequence, and it picked up it picked up what was needed to be expressed? I think there's a great deal of hand-waving that goes on in the literature about the origin of orphans from non-coding sequence. And let me put it this way. Even if you have non-coding sequence, you run into a problem because the total nucleic acid debt has got to be paid as you go back in time. I can't express it more precisely than that, but you end up having enormous amounts of nucleic acid necessary to account for the total number of orphans present, and it really doesn't solve the problem. Anyway, Anna, in the next year, I'm going to be looking into that in greater detail and really, trying to analyze some of these models.
Okay, thanks.
So Roald Truman also said that we also find taxonomically restricted miRNAs and transposable elements.
Yes, and in fact, I just saw a really nice review paper on transposable elements. Transposable elements turn out to have very, very strange distribution patterns, and they appear to be species specific. So, what I looked at with bugs are mainly protein coding sequences, but the same pattern of taxonomic restriction applies to non-coding elements as well. And so we're in for a wild ride.
Salvador Cordova wanted to know if orphans are also identified by RNA transcripts or DNA gene prediction. They're primarily identified by DNA sequence that is put into gene finding programs, and then the output of the gene finding programs is just fed into BLAST, and you go out into the databases looking for hits. When you are analyzing, of course, the functional role of an orphan, you're going to be looking for mRNA transcripts, you're going to be looking for expression. As some of you know, in 2007, in Eric Lander's lab, a group ended up dumping almost 1,200 human-specific open reading frames because they said they didn't have RNA tags, and really not a very good analysis at all, because if orphans have very, very specific functional roles, they may be present as an mRNA for only a very brief window in the life history of an organism. And if you haven't sampled at the right moment, you're not going to catch the express sequence tag that you would need. So another project that I'm involved in at Discovery is going back and looking at that paper. The lead author is Clamp, to reanalyze what they did, because they dumped out a significant fraction of the human genome because it was found only in Homo. So you also have Walter Remind wants to know if taxonomically restricted genes exist between organisms that are known to be able to interbreed, such as between dogs and wolves?
That's a question I can't answer because Bugs and I are looking at, in the case of dogs and wolves, that would be in the genus Canis. So the analysis for us would be at a higher taxonomic level than the single genus. I know Walt's concern is that we not use orphans to diagnose designed or created groups because you've got hybridization with fertile offspring, and if you're using orphans as your diagnostic criterion, you may end up making a mistake as you're trying to diagnose your designed sort of aboriginal groups. It's an important question, and I want to look into it, but I haven't yet.
And if I understand what Royal Truman is saying, is that the chances of new enzymes like topoisomerases and methyltransferases, the chances of having new ones that completely mess up a genome as opposed to help it is way greater than 99%.
Oh, I agree completely. And one of the key things about essential hardware is I discovered this when I looked at theories for the evolution of the genetic code. In every case that I know where the code has been experimentally modified, where you're removing a codon, you're removing a transfer RNA, let's say, completely from a genome, you have got to provide the wild type representative on a plasmid or in some other way to bridge the functional space as you're changing essential hardware. So the reason I think Change is really onto something is she realizes that essential hardware is not something you can just swap in and out willy-nilly. You have to provide a mechanistic pathway to get from A to B, where that job is being done by something. I think that these dual criteria of taxonomic restriction and demonstrable essential roles really does give us a tool to diagnose what may be the original design groups.
All right, does anyone have any other questions?
Let me say, if we do this again in a year, I hope to have some interesting results to report, and I'll be back even more animated and jumping up and down.